In this presentation, I will talk about electron microscopy for the determination of protein structure. I will start with an introduction of electron microscope. In comparison to light microscope, electron microscope use electrons for illumination. As you can see here in this picture, through the electron gun, the electrons are generated, which are traveled through the electron column, and they strike at the stage where the specimen is present. So at this stage, specimen can interact with the electrons. Either it can scatter the electron, or electrons can be transmitted to the specimen until they reach the stage where there is a detector and these electrons are detected and information stored in the sample is generated. So electron microscope can be divided into three parts. First is illuminating system, the second one is specimen plane, and the third one is imaging system. Electron microscopes can be used for studying the ultrastructure of biological molecules, inorganic specimen, microorganisms, cells, molecules, metals, and crystals. Electron microscopes are of two types. First is transmission electron microscope, which is used for the determination of protein structures, and the scanning electron microscope, which is basically used to scan the surfaces of the molecules and microorganisms. Now I will differentiate between scanning electron microscope and transmission electron microscope. So in scanning electron microscope, the type of electrons are scatters which are detected. In a scanning electron microscope, you use low voltage as compared to transmission electron microscope. The specimen thickness is really thin in transmission electron microscope because the electron need to transfer through the specimen, so it should be really thin. In scanning electron microscope, you see the surface structure of your specimen or sample, whereas in transmission electron microscope you see the 2D projections. The magnification is higher in transmission electron microscope, whereas the field of view is larger in scanning electron microscope. The optimal resolution for sure is better in transmission electron microscope, and the image formation is in scanning electron microscope, information is through the detector from the electrons which are captured. And in transmission electron microscope, there is a CCD camera which can detect the information stored in the sample. About the operation, scanning electron microscope use little preparation of sample as compared to transmission electron microscope, which has to be really heavy preparation of sample. There are two different methods for preparation of sample for electron microscopy of protein structure determination. First one is negative staining and the other one is cryofixation. In negative staining, you add the sample with buffer on a carbon coated grid and you blot the sample with the blotting paper, and then you add heavy metal stain and you blot again. As a result of this arrangement, the protein molecules are attached on the surface of carbon, which are air dried, and these molecules can be studied with electron microscope. Whereas in cryofixation, the sample preparation is bit different and bit complicated. Here you can use the holy carbon grids. In the holy carbon grids, you apply the sample and protein is entered in these holes of the grid. Then you blot the excess sample and you quickly freeze the sample in liquid ethane. As a result of this, protein molecules are frozen in the thin layer of ice, and you can see them through the electron microscope and determine their structure because they are randomly organized in the thin layer. This is the workflow of electron microscopy for 3D structure determination. For sure, the first step is the isolation of photosynthetic supercomplexes. After the isolation, you apply the supercomplexes on a grid, which are transferred to the microscope, where you can record several thousand images after screening the grids. So from all of the images, you select the particles, you pick the particles and you extract them. After extraction of particles, you align them in 2D classes, and from those 2D classes, the best classes are selected for 3D and 2D structure determination. Now we'll talk about single particle analysis. For this, you record a few thousand images and you select, as I said again, the best images based on CTF correction, and then you extract the particles and then you align the particles into 2D and 3D classes. Afterwards you get the 3D model which is refined and post-processed, and afterwards you model your structure. This is a typical example of an electron micrograph, and you can see the protein molecules. This is the top view of photosystem 1, again top view, and this is side view, and this is the tilted view of photosystem 1. And these are the 2D classes of photosystem 1: side view, top view, and tilted view. Cryo-electron microscope has really advanced in a few years. After 2013, there was a rapid advancement in this technique after the development of new cameras, softwares, and the generation of new methods for separation of particles. So now it's possible to reach near atomic resolution using electron microscopes. In 2017, Jacques Joachim and Richard got Nobel Prize for the development of this method. In this slide, I will talk about image processing. So from the raw images, you collect images something like this. From these raw images, you sum up the particles on top of each other to get 2D templates. From these 2D templates, you can generate 3D initial model, and you can also align and group these 2D classes into 2D averages, which show better features of the particles. As you can see in this picture, it looks like original one. And afterwards, you can reconstruct the 3D model from 2D classes. Again, I will explain with an example the image processing. So this is the raw image, and you select the individual particles from raw images. You extract the particles and you sum all of these particles. For example, this is sum of 4, 8, and 32 particles, and more particles you sum together, better the resolution gets. So you sum up thousands of particles together to get all the views of a protein molecule. For example, top view, side view, and tilted views. So this is our initial model of photosystem 1. You can see the core of photosystem 1. This is the core, and these are the belts of antenna proteins around photosystem 1. I can start the video to show you the 3D structure. Okay. So now you can see the stromal side of photosystem 1. This is luminal side of photosystem 1, and you can also see the iron sulfur cluster in this photosystem 1 structure. Thank you for listening.